Okay, hello to all of you. So let's discuss the Tetrahymena lab. So basically, Tetrahymena is a non-photosynthetic single-celled eukaryotic organism. So what we are trying to study in this organism, we are trying to study phagocytosis. So phagocytosis is a kind of a endocytosis in which the cells, they can engulf something solid. They can take in something which is solid. So basically, in this lab, if we would have done this lab in the laboratory, we usually use this organism called Tetrahymena, and we mix it with latex beads or India ink. We have been usually using India ink because this India ink actually goes inside the cell of the Tetrahymena, which is a single cellular organism, and it goes and basically binds to these, or it labels all these the food vacuoles inside this organism. So the more the phagocytosis, or more this organism is feeding on, the more vacuoles you see um, inside. So basically, in this lab we usually use a phase contrast microscope. So we focus this organism and we try to see the vacuoles which are labeled with the India ink dye inside in, inside the Tetrahymena. So basically, let's first go over the methodology, what you would have done if you would have done this lab in the lab. So yeah, so basically the aim of this lab is to determine the feeding rate in Tetrahymena. So in this, as I said, we mix the Tetrahymena organism with the either the latex beads or India ink, and then we let them be together for some time, and then we take a slide. On the slide, we take a Lugol's dye, which is basically iodine, and then we add this mixture of the organism and the India ink on the iodine, and it basically kills the Tetrahymena, and then we put the cover slip and we can see these vacuoles, the food vacuoles inside the Tetrahymena cell. And we do this for different time periods. So this is basically the methodology. So very simple methodology. You mix these Tetrahymena and the India ink, and then you take them on the slide. On the slide you have a Lugol's reagent, which is basically iodine, and then you look under the microscope, and you do it for different time periods. Okay, so so basically, see, when we are trying to determine the feeding rate, as I said, I'm also going to show you how basically we do. I have already provided you the Excel sheet, and the Excel sheet, the data is already there, and the calculations are all already done. So basically, sometimes when we do the experiment, we can get some incorrect results. So sometimes maybe the Tetrahymena has actually stopped feeding, but sometimes still we get the false results, so which the results indicate that the Tetrahymena is still feeding. So so there can be a chance, there can be some experimental error, because of that we can get these kind of results. So to prevent that, we are going to learn in this lab how to do the statistical analysis, and how to figure out on the basis of the statistical analysis if the results which we are getting are correct or not. So there are two kinds in the when we talk about the statistical analysis. So we have two kinds of hypothesis. One is called the null hypothesis. Another is called the alternative hypothesis. So the null hypothesis is always a hypothesis which says that whatever we are trying to study is not going on. So it's always opposite to what we are trying to study. And alternative hypothesis is which basically says that yes, there is an effect of something or what we are trying to study. So null hypothesis is always opposite. So in this experiment, in this specific experiment, the null hypothesis will be the feeding rate of Tetrahymena is that the Tetrahymena um, they the with time the Tetrahymena they are not feeding. So that is a null hypothesis in this case, that the tetrahymena, they're not feeding as the time is passing. So the feeding rate over the time is not increasing. That's a null hypothesis. But the alternative hypothesis will say that with time the feeding rate is increasing. So that's going to be the alternative hypothesis. So basically, methodology, I have already told you what the methodology is. So this is all. You can go over here, everything is written here. And what you have to do here, if you come to the Excel sheet, so the data, so as we look for the feeding of the tetrahymena over different time points. So first of all, we look for 5 minutes, then we go for 15 minutes, then 25, and then 35. And this is the data given for the beats per time. So which means that, and also when we do the experiment, at every time point, 5, 15, 25, and 35, at every time point we go and look for 8 tetrahymena cells. And in so we go and look for 8 tetrahymena cells, and we go and count the these vacuoles inside each tetrahymena cell. Or we can also use the latex beads. So the latex beads, they actually go inside the tetrahymena cells. I have also posted one PowerPoint in which you can see actually the pictures, how the tetrahymena would have looked under the microscope. So these are the values. So which means if you're using India ink, like at 5 minutes, if you go and look for 8 different tetrahymena cells, this is the data. So in the first one you see 2 vacuoles, in the second one you see 4, in the third one 3, 1, 0, 8, and this is the data for 15 minutes, this is the data for 25 minutes, this is the data for 35 minutes. So basically when you add the data in this Excel sheet, you get here some values. Now what is the M value? M value is basically the feeding rate. T value is a statistical value, and DF is degrees of freedom. Degrees of freedom is usually, like, this is around 32 data points. So degrees of freedom is usually -1 the number of values you have in the data point. So we have around 32, so just subtract 1, so it becomes 31. So we have 31 degrees of freedom, this is the T value, and this is the feeding rate. So now using these values, we have to figure out if the tetrahymena, their feeding rate is increasing with time or not. So how to do that? So what you have to do, you have to look at your T value and you have to compare it with another value, which is another this table I have already given you. So if you look here from this table, you are going to figure out the critical value for your experiment. And how to figure out the critical value? For the critical value, what you have to do, on this side, if you look here on this side, these are called degrees of freedom and these are called the probability values. So you have to look on this, we are using t-test 2, so you have to look at these values. Okay, so what you have to do, these are probability values. So we are going to look for the probability value of 0.05. You are going to look for the probability value of 0.05, and you are going to look against the degrees of freedom. So in this experiment, the degrees of freedom is 31. So this is 31, and you have to go and look under 0.05. So this is 0.05 probability value. You're going to look under 31. So the value is 2.040. So this is your critical value. Okay, 2.040 is your critical value. Now what you have to do, you go back to your data, you're going to compare this critical value with your T value. Now important thing to remember here is, if your T value is bigger than the critical value, then you are going to reject the null hypothesis. So the null hypothesis is always opposite to what you're trying to study. So the null hypothesis in this experiment is the feeding rate with time is not increasing. Okay, and the alternative hypothesis is the feeding rate is increasing with time. So your critical value is around 2, right? So your t value is 1.29. So in this case, your t value is smaller than the critical value. Okay. So now, as I said, if your t value is larger than the critical value, then you're going to reject the null hypothesis. When you reject the null, accept the alternative hypothesis. But in this case, t value is less than the critical value. So it means that in this case, we cannot reject the null hypothesis. So which means that we have to accept the null hypothesis. So accepting null hypothesis means that the feeding rate of Tetrahymena is not increasing with time. It's not increasing with this time. So with time. So this is basically all what you have to do. So in the lab there are some questions. If you look here, there are 5 questions. These are the questions you have to answer. That's all you have to do for this lab. Okay, so you do not need to fill this part because everything is there already in the Excel sheet. I've already provided you with the data. So these are the 5 questions which you need to answer. So if you have any question, you can please let me know about it.